Hello students, this is Dr. Faizan Mirza. As we already have covered how a monoclonal antibody can be produced by injecting an antigen, the desired antibody can be produced inside a mice, the lab animal. And how is that possible? You just inject the antigen against which you want the antibody to be produced. So once the antigen is injected into the mice, then the hybridoma cells can be produced by fusing the plasma cells from the spleen of this animal and the myeloma cell, the cancer cells, and you add fusogen or electric shocks to make sure that the two cells fuse together. The cancer cell provides the you can say the ability to the plasma cells to keep on multiplying infinitely, and we know that plasma cells do not divide, they are short-lived cells. So plasma cells will have the ability to produce the antibodies against the antigen that was injected, and the cancer cells will provide the infinite ability to keep on dividing. So the hybridoma cells can then be grown in lab, and they will keep on producing single monoclonal antibody, which can be used in treatment as well as diagnosis. We have already covered how its treatment is, how trastuzumab is an example of a mAb, a drug which can be used for treating the breast cancer, where overexpressed antigens on the breast cancer cell can be marked using the antibody. Because its variable region will be complementary to that antigen, it will go and mark the breast cancer cells. Similarly, deep vein thrombosis can be diagnosed using a monoclonal antibody which has been raised against the fibrin antigen which was injected in the mice. How the application of mAb differ? So we know when we are using monoclonal antibodies, we know that the main purpose of using mAbs is that it speeds up the process of treatment as well as diagnosis. For diagnosis, it is administered as a single dose, but for treatment, it is given as a regular administration for a long-term outcome, as well as follow-ups are kept in check, so we know the progress of the patient. Since the antibodies they come from mice, they can trigger immune response in the patient's body, because they will serve as non-self antigen and they can trigger an immune response. This needs to be resolved. It can be resolved in two ways. Number one, genetic sequence for the heavy and the light chains of the antibody from the mice can be altered. They can be changed to code for human chains of amino acids which have the R groups, or you can say which are having those amino acids which are exactly like the constant regions of human antibodies. Similarly, the carbohydrate which is attached to the heavy chains, it is changed. From the mice, it is changed, its position is changed, its position and type is changed, so it ends up resembling the one which is present in humans. As a result, the mice antibodies do not look like mice antibodies anymore to our immune system. When these are injected, mAbs, when they are injected either for diagnosis or treatment, they do not trigger an immune response in the patient, whether it's used as for the diagnostic purpose or for treatment. That's it from my side. Thank you so much.